to finish up the superior colliculus. We didn't quite finish last time. Here we have a midline and the superior colliculi, the pineal gland superimposed on it, the inferior colliculus below it. So this is a view from behind. And what was found just about 50 years ago by Julie Apter was that the retina projected onto the colliculus with visual field looks something like this. This is the center. And then if she stimulated the colliculus, she got the eyes to move conjugately to the point to which the retina had projected. So if this was up, and this was down, and these were increasing degrees of left conjugate gaze. And this fitted very well with the idea of Perronneau's syndrome, which most commonly occurs in cases of tumors of the pineal that could press bilaterally on the upward conjugate gaze region and produce a paralysis of upward gaze. And that seemed to fit very nicely clinically, except when Bender's group began to study animals, particularly monkeys, and would take out the colliculus, and nothing happened. So that all of the nice retinotectal and tecto-conjugate gaze connections that have been demonstrated for superior colliculus didn't seem to have very much actually to do with the colliculus. You could take the colliculus out and have no conjugate gaze quality difficulty. And so the situation has been rather cloudy until a few decades ago, I guess, when it became clear that the descending cortical, if this is in midline here, that the frontal eye field, for example, tracks would come down and along with the other cortical bulbar, cortical spinal tract fibers, but take off from the internal capsular fibers and come along the walls of the third ventricle. The others going down sort of face, arm, and leg out here in the end of the capsule, and then down the peduncle. These would come down the walls of the third ventricle and cross somewhere in the region of the posterior commissure and descend on the other side of the brainstem as the conscious conjugate gaze mechanism. And this we saw a week or so ago when we were talking about the NLF and the vestibular intersegmental connections. This being a tract in the brain stem is just a few millimeters lateral to the NLF and has a quite different function. The lesions that we've seen in this system have involved the walls of the third ventricle bilaterally in patients who had a basilar artery insufficiency and infarction of the medial thalamus and a loss of conjugate vertical gaze. And so it's thought that most of Paranoid syndrome with the pinealoma relates to damage to these fibers as they're crossing somewhere in the region of the posterior commissure. So any questions on this little piece of the non-functional superior colliculus? Paranoid syndrome does not seem to result from collicular lesions, even though the colliculus receives from the retina as retinotectal fibers into what layers, Eric? Outer two layers, stratum zonale, stratum opticum, coming from retina on the one hand and occipital cortex on the other. And exiting from what layer, Linda? How many layers of gray matter are there in the supracolliculus? But you know the answer is 3, of course. And so it's the deep layer that gives rise to the exiting fibers, which would form the crossing of the tectobulbar, tectospinal tract, and collicular, more laterally directed fibers into the forebrain. Those connections do not seem to be involved in Paranoid syndrome. So any questions? In this regard, let me just mention something that I left out of the cerebellum. You'll recall, I hope, if we take the cerebellum here and set it out, with the vermis and the paravermis lateral cortex, oculonodular lobe, that there is in the anterior vermis a tail, leg, arm, head projection of spinal and trigeminal afferents, and in the paravermis a similar one, but the other way around, tail, leg, arm, and head, bilaterally. It says that this central region contains projections of the head, including not just somatosensory and spindle, but also auditory and visual. And while it's not really known how these two special sensory systems, auditory and visual systems, project there, it is, let me see if I can get the colors right here. The tectum projects down to a nucleus of the pons. Colors. Over to the deep layers of the tectum, going out, crossing, coming down to the pons, the dorsal lateral region of the pons, basis quantus, that is, and then fibers crossing come into the cerebellum through the middle cerebellar peduncle into the central region where head fibers should project. And so we have a possibility of corticotectal, tectopontine, pontocerebellar, or visual system, from cerebral cortex to cerebellar cortex, from retinal afferents to colliculus to cerebral cortex, and presumably, or at least possibly, if they want that tectal connection, into the pons and cerebellum. I don't know if anyone has actually traced those physiologically and severed them to show that they do take that path, but at least that's one path that's available. So one can stimulate in cerebellar cortex and receive in cerebral cortex, or vice versa. We can stimulate the retina and project occipital cortex to tectum to cerebellar cortex. So we have, and similarly for the auditory system, we have the peripheral special sensory systems projecting to cerebral cortex, occipital or temporal lobes, the cerebellar cortex, about the same central region, and to the two colliculi, one visual, one auditory. But the interconnections between those are not, to the best of my knowledge, proven. But it did come as quite a shock some decades ago to realize that the cerebellar cortex received these special sensory inputs. And even more, that they were interconnected, cerebellar and cerebral cortex. So any questions on the colliculi, on the cerebellum, on the segments?
Just one minor thing. First part, the corticotextal fibers, is that not crossed?
Well, they should be crossed. What do I got here in my notes? Well, my note shows them uncrossed. I have to stop for a moment to think whether they should be crossed or uncrossed, because it would be nice to have the visual field projecting to the optic colliculus, and to have the cerebral cortex connected to the optic cerebellar cortex. And now to get that connected up properly between one colliculus and the cerebellum, if we come down uncrossed to the pons, and then crossed at the pons level, between business. So that's probably the way it works. Thank you. Any other questions?
I was wondering if you could explain how cerebellar muteness occurs.
How?
Cerebellar muteness?
Cerebellar
Muteness?
muteness.
Mutism?
Don't know anything about it. Tell me about it. We'll take a
We
crack
just
at it.
had somebody with a cerebellar hemorrhage at the VA that became mute. I guess it's well described in children with tumors, but in adults it's fairly infrequent.
I temporarily at a loss for words because I haven't heard about it before. These would be with acute lesions or with tumors or?
Actually, this was an acute lesion, and actually over a few months it resolved. It was left with cerebellar speech.
I can understand the resolution easier than I can talking about. Because in general, cerebellar lesions symptoms do resolve in a period of months. Particularly if it's out in lateral cerebellar hemisphere.
Actually, this was midline vermis.
Midline vermium lesion. No, I just have to mix something up.
Yeah, in kids, if they split the lower vermis, usually the lower part of the vermis, lower third, that, like for a posterior fossa tumor, they wake up mute, and it can be very profound and last for, weeks. And the only other thing I can think of is that when from PET scanning, that when they do PET scans, they usually some contralateral midline-ish just a paravermia kind of thing lights up on PET scanning with the language test. But why? What the connections are to cause that?
So you're splitting the vermis posteriorly, but how high up?
Third to halfway up.
Doesn't seem quite like it's getting up into where all the other head functions would be located. But you might be encroaching upon it. On the afferent side, on the output side, I don't see any particular connection back except through vestibular roof nuclei, stage 1. No, I don't see any ready explanation. If it were up here in cerebral, in the head areas, central area of the cortex, I could see that it might be interfering with a lot of the coordination of your speech through second stage of cerebellum, and that it might take some considerable while before that incoordination so severe as to manifest itself as doing nothing would resolve itself and be able to do something with a very awkward, incoordinated inability to control rate, pitch, volume of speech. On short notice, that's as close as I can come. Any other questions? Well, David keeps pressing me to give you an exam. And so I thought instead of an exam, we'd have a review session in which we would look at the contents of the brain. Question?
No, we're just thanking David for continued support. Where he lives.
And the anterior hindbrain. Middle hindbrain. And the posterior hindbrain. And I guess I'm going to put some spinal cord in there. Because we've come to a point where from now on the systems get bigger. The forebrain all the stuff that lies above the aqueduct, above the cliculus, around the third ventricle, around the lateral ventricle. Those structures are going to get bigger and bigger. And so they're pathways that get down here in the brain stem are going to get bigger and bigger. And these smaller structures we've been talking about for the past month, almost a month of July, are relatively small. And if we don't pay attention to them, we might lose them. So, let's see how an analysis of segmental and the first two suprasegmental organs can fit into cross-section at various levels. So the exercise is one of converting all that I've given you so far, segmental, suprasegmental, into cross-sections. It's the reverse of the way I was taught 50 years ago, of looking at a cross-section and learning everything in it. And of course, in theory, once you know all those, you can work it either way. But for my basic plan in this particular course is to show you how to build it up, segment by segment, and suprasegmental afferents to segmental efferents, and let's give it a try. So what are we going to find in this cross-section of the midbrain? Where are they going to be? Jonathan, want to start?
So what, just in terms of specific
Well,
tracks?
how would you approach it?
Well, there's
I've given you an outline of the section. That you should have to remember, I guess, to begin with. You could, I suppose, build it up and end up with that outline, but this is the easier way to do that part. Now, what's going to be in this cross-section?
okay. Well, most ventral are cerebral peduncles.
Okay. I was hoping you wouldn't begin there, but we have to put some names to these structures that we give. So we have the peduncles here, and it's changed the nigra here, and the tegmentum here,
Part of follicula back there.
and the tegmentum in there. Sort of dividing line.
Aqueduct,
So
periaqueduct.
those are in the aqueduct, in the periaqueductal gray matter.
That about covers it.
Those are the gross structures that we have to play with in this section. Ray, what would you like to add? Now we've done the hard work.
Is the red nucleus in that section?
The red nucleus would be in this section, yes.
In the tegmentum?
And it's going to be here, above the suspension nigra, about in here.
If you want to think about
Now you're sort of doing it in a non-logical fashion. I mean, there's a certain logic to what you're doing, in the sense you're taking the big structures first.
well, I was just thinking that if we wanted to take suprasegmental structures and kind of draw them down, and we've got the tectum on there, and I guess that's going to send things down to the spinal cord.
Okay, so if we take the layer 3 and send its fibers out and across the other side, this tectospinal, tectobulbar, tectospinal fibers, they would be running down to the other segments, sure where they are in the spinal cord. Okay. This is running the course backwards now, taking the most recent things that we've done. So that's legitimate.
Okay, I wanted the red nucleus on there because that, I guess, gets from the other suprasegmental thing we talked about, the cerebellum.
Okay, so we have that coming from, okay, we've got a cerebellum here. So we have the dentate nucleus. Giving rise to fibers that go out, crossing, coming into and around the nucleus on their way up to thalamus. Okay. You want to put the rubrospinal tract in too, as long as we're here?
Yep.
Okay. Crossing. Rubrospinal. Not sure where the hell the rubrospinal tract is here. It's supposed to be somewhere out here in the spinal cord, in front of the spinal tract. But just where it is in the rest of the brain cell, I've never been sure. So, trying to keep track of where we're going here, and backwards.
Okay, we're just adding on tracts that are efferent tracts. Put the cerebral peduncle on there, but then we can mention that there's...
Well, we got the peduncle here already grossly. We'll come to that, to its components, another day or two.
I think those are those suprasegmental tracts that originate there. There are some segmental things that originate there. You want to switch to those?
But if we're going to do this backwards, we probably ought to have some deep nuclei here of the intermediate. We're also going to give rise to crossing fibers of the superior cerebral peduncle after that nucleus back down into the spinal. This is a challenge to do it this way. Mike, what will be your next?
Well, I think that would be a challenge also. I think the easiest way would be to do it from a segmental approach, build that up so you can look for those things and then add in. That's going to add in things.
I think that's the easiest way to do it. Begin at the beginning and put the segmental structures in.
And I put segmental structures on each of those segments.
Because it's difficult to put the intersegmental connections in unless you have the segmental. And it's difficult to remember all of the suprasegmental structures, particularly on the afferent side, until you have the association plate. So doing it this way and starting at the end, trying to work it backwards, forces you, I think, to go back to the beginning. Now, it's easy with a computer, it can do it like that. But with our own memory drum, it's more difficult, I find, to run it this way. Follow the cerebellar pattern of taking the third stage efferent, the second stage efferent, your logic in the first stage efferent, which requires that you have a vestibular nucleus, goes as well as receive from. And if you're not careful, doing it in some kind of systematic fashion, you leave out a piece. And having left out a piece at one corner, it becomes difficult to ever get it back in. So, Mike, let's go back and try the segments.
I would have said with the greatest possible due respect, I would rather start over here from the built system on Wednesday.
Here.
Yeah, start at the bottom. And that
All
way.
right.
I'd start with motor. Start with motor.
Here's your horn cells. Medial lateral,
And dorsal lateral.
and dorsal lateral. Postganglionics. Okay. So that's basically our motor plate.
And just like
Oh,
on
spinal
spinal
cord.
cord, I remember just to put a gamma in there somewhere.
And we got to put a gamma element in here too. Okay.
I'm going to do my sensory plate segmental structure.
So I've got
Small in temperature system coming in with Sauer's tract and then median and then large.
okay. And now, Eric, what's next?
I feel the association plate.
Sorry.
The association
Right.
plate.
Okay. Which would like?
The small one, small peak temperature, we have 3 nuclei.
Marginal to that less appropriate. Okay. And the output appropriate to.
It goes back to some of the alpha motor neurons and has some crossing fibers to the lateral spinal cord.
Okay. And also to. These are going to go to flexor motor neurons. Ipsilaterally. And contralateral extensors. Basic cross extensor flexor withdrawal reflex built in. And here we come out to the spinothalamic tract, which is going to be out here in this diffusely anterolateral quadrant. Lateral spinothalamic. Okay.
Right now, the minute I get one of those, I want to run it up. So,
All right. Where would you like it up here in the next one? Eric, where would you like the spinothalamic tract in the next higher segment?
Stays lateral.
ready.
So they're just above the olive, out laterally. And next higher. Okay, next. Next. So small fiber system. Polysynaptic. Put all the synapses in here. But the final output, which really comes to about maybe 10% of the system. Most of it being used up in segmental, intersegmental connections. About 10% of it actually reaching up the thalamus. Going through these cross sections or past these other segmental structures on their way up to thalamus. And giving rise to collaterals that get off into the pecten. Coming into the stratum menisci, somatosensory afferents into the pecten. The collaterals into reticular formation. Okay, who wants to do the next little piece? Linda? Want the medium-sized fibers? All right. What are they going to do now? Medium-sized fibers are not going to go to Clark's nucleus. Something close. They'll go up, push your columns. Now, at a level below this, down by the obex, so I better draw a slightly different piece of medulla here. Not very good at drawing this next intermediate piece of medulla. We have to put in some central gray in the midline and rascal pineatus trigeminal, inferior olive pyramid.
Good job.
Something like that. And so we have the posterior column fibers coming in and ascending and synaptically on nuclei posterior columns. Easel here, so we have. And what happens now? We left them there last time, didn't we? They're going to come down and cross into the medial meniscus, which is going to rise here just above the pyramid. Get pushed up and slightly laterally, next here, into the junction between tegmentum and basis pontus, medial to the spinotonic tract fibers. Also giving rise to collaterals like this. Also giving rise to collaterals in the reticular formation. But basically on this way up to the thalamus for conscious projection, for projection of conscious medium-sized fiber systems, which includes essentially everything except any temperature, spindles. And what was this little system down in here that Linda started on? This is not Clark's nucleus, but closely associated mnemonically anyway, because it gives rise to the bilateral system, the ventral spinal tract. Which, now, where is it in these other regions? I'm not honestly sure where it is in these other regions. It's lost pretty fast, except that it comes up here into your anterior hindbrain. So it's in somewhere here, close to main sensory nucleus, and entering cerebellum through the superior cerebrocanalicle to take with the sensory nucleus of the trigeminal the old primitive spinal and trigeminal cerebellar infill for everything except pain temperature and spindles. So we have that dichotomy of functions, dichotomy of projections for all the various functions of medium fiber systems. One conscious, medial meniscus system. One unconscious, cerebellar. Okay, how about spindles? Rita, how are you in spindles? Large fiber system.
My name's Petra. My name's Petra.
I'm sorry?
My name's Petra.
Oh,
Not Rita.
sorry. How does the spindle system work? Now the synaptic connections down to optomotor neurons for stress reflexes. Stretch the spindle, evoke a contraction. And now we have to put in another piece of spinal cord. Well, we can put it in the ideal section. I'm running out of color. Another color. Now we need parts. This is the one that's going to receive the other collateral of the spindle system. Generally not in the same section, but a corresponding segment rostrally, it's a lumbar segment, caudally, it's a cervical segment, because Clark's nucleus is depressed in the thoracic regions. But on a segmental basis, still cervical, sacral, just kind of scrunched down together from T1 to L1. And then Clark's part, just over here is the dorsal. Spinal cervical tract. This comes up into the inferior cervical peduncle, coming up and into the cerebellum to present the cerebellar cortex with mossy fibers, granule cells, Purkinje cells, and now they come to the second to go to this stage that can be brought up. And then we need the equivalent of that for the trigeminal, for the head system, which is going to be, put that on the same side here, out of here, right over the aqueduct, there's the spelling, trigeminal nucleus, its suprasegmental part, remembering that the segmental portion of that nucleus are subdivided into cell bodies whose fibers must make connections somehow to the association plate element, project into the cerebellum, again through the superior cervical peduncle, and those spindle afferents will be coming out through the anterior hindbrain segment here. Now, down, we'll have spindles with the third dimension. Trigeminal for jaw muscles, through first division for periorbital muscle spindles, and however else, other head spindles.
How about going on the arm component of that, like lateral
Thank you. So now we've got to put in the ascending collateral up to the lateral cuneate nucleus. That'll be coming up here. And lateral cuneate, I want to put in purple. Cuneate's going to be here, so lateral cuneate's going to be here. And the fiber is going to come into inferior peduncle with the others. into the cerebellum. We have lateral TH. Cuneate, wrestle there.
So is the lateral cuneate in that section really right next to the rest of the dorsal
Right.
spinal fluid?
Right next to the lateral cuneate. So the segmental spinal cord is reasonably complete. We haven't completed we've done the spindle system, we haven't done the medium and small fiber systems for the other brainstem segments. The easiest of which now is going to be the small fiber system. Because here we have coming into the side, small fiber system, trigeminal, branching, running, tract, shooting out collaterals, running a tract, laterals, joining the cervical segment of the Sowers tract, giving out secondary collaterals to the same kind of laminated nucleus that lies in the caudal third of the system, so we'll put it mostly down here. Giving rise to crossing fibers come up to join the leg and arm fibers crossed spinal tract fibers. So they'll be running up here, leg and arm. They're coming in, crossing rather obliquely, and come in here and run on up. And that's going to take care of essentially all the pain temperature system for all the head segments. Whether it comes in through anterior hindbrain, or middle hindbrain, or posterior hindbrain. So we've taken the easy ones, the small fiber system and the large fiber system, because they are almost identical in spinal cord and brainstem segments. Or very reduced for brainstem segments. But have very analogous components we kept track of. The equivalent of spinothalamic, spinocerebellar, trigeminothalamic, and trigeminal cerebellar. And that then leaves us with only the medium fiber system to worry about. Medium fiber systems, essentially everything else. We've taken care of them for the spinal cord segment, posterior column, nuclei posterior column, and the deep nucleus for the ventral spinocerebellar tract. But they become much more complicated, much more specialized when we get up into brain stem sections. And so, we need to put some motor and sensory plates in for each of these segments. Otherwise, we have no orientation. So we've got a medial groove, 12, a ventrolateral groove, the 9th, 10th, 11th. Remembering that that's cervical. Same ventrolateral nucleus though. And a dorsolateral groove for 9 and 10. Coming out medial to sulcus limitans. Next higher segment, we have a medial groove. Down to the 6th. A lateral groove. Laterally to the 7th. And a dorsolateral groove. For ganglionics. Anterior hindbrain, we have a lateral groove. That's all. In the midbrain, we've got at a lower level the fourth nerve, which is curious course. And a third nerve nucleus. There's medial motor neurons and lateral ganglionics. So we get these small structures in place. And the sensory plate with small and medium-sized fibers. Ganglia, or posterior hindbrain. Small and medium-sized fibers for the middle hindbrain. We put some small ones in already, it looks like. These some medium-sized fibers for the trigeminal. And except for mesencephalic trigeminal, where it's put in, we don't have any other small or medium fibers in midbrain. We've taken care of the association plate. There's small fibers, spinal cord, small fibers, posterior hindbrain, small fibers of middle hindbrain, small fibers of anterior hindbrain, because all of those come into the spinal tract of the trigeminal and are descending to end in that laminated nucleus and temperature. We've taken the large fiber system where we know it, for spinal cord, midbrain, and now I have all these others. So all the other kinds of sensation for most vital reflexes of posterior hindbrain, heart, lungs, and GI tract, vomiting reflex, blood pressure reflexes, respiratory reflexes, and all of those come into the solitary tract and its nucleus. Now the lateral to the sulcus limitans. nucleus sort of stands out. And then the projection of that nucleus outside of the segment is not well known. Most of these connections are made to reticular formation, posterior hindbrain region, or reticulospinal connections, or for intra-segmental connections. The rostral part of that projects up to thalamus and ventral cortex for conscious appreciation of taste, but just where those fibers run in the brainstem, I don't know. Medium-sized fibers of the anterior hindbrain with the main sensory nucleus crossing to join those of the medial lemniscus. And so pretty soon we're down to this relatively complicated middle hindbrain segment and its vestibular cochlear components. And that means that we have to find a place for those nuclei. That are going to fill in the space and overlap here. Don't go down this far, but don't go up the anterior hindbrain. So we have the vestibular nucleus and the bipolar vestibular afferents and the cochlear nuclei. Well, put it here. Dorsal distal cochlear nuclei. And the bipolar again to end on these two association plate elements. Now we can put in the intersegmental connections, vestibular. That's the MLF. Connecting all of the extraocular muscle nuclei of midbrain, running through anterior, including middle hindbrain, posterior hindbrain, eye and neck muscles. Having gotten that into its primitive subependymal region, we can then add in later newer stuff like the tectospinal tract at the start. Can't go quite that high because it's already got something more primitive in its way.
Is there any difference between the dorsal and ventral cochlear nuclei other than dislocation?
Functionally, not that I know of.
Are they really two discrete things, or is it really all just one group?
No, they look quite different. Ventral, very really large, closely packed cells. Dorsal, much less so. Giving rise to fibers that partly cross, partly don't cross in the trapezoid body and ascend in the lateral meniscus. Not quite this high, but go up into the inferior like this, the lower levels. So the lateral meniscus out the surface. Medial meniscus down below. So we've begun to fill in much of the space by a relatively simple device of getting the segmental structures in place, and then projecting them into cerebellum and colliculi for each of the association plate elements, which there should be 3 for each segment. There's small, medium, and large fibers. We left out a big system here, and that's the input to ocular nodular lobe of stage 1 cerebellum. When we came to the posterior nucleus, we got the intersegmental, but forgot the main suprasegmental connection. We didn't emphasize, but remember this map, you know that the spinocerebellar tracts come in and the trigeminal tracts come in in a laminated fashion, horizontally. And the output going saggitally through deep nuclei. First stage of cerebellum, vestibular. Second stage we put in here with the intermediate nuclei, going out to red nucleus, rootless spinal tract crossing back. Third stage of cerebellum, dentate, red nucleus, thalamus. And so we got the major afferents and efferents of cerebellum. In the colliculi, we have the inferior colliculus going up to the medial geniculate. Then we have an input, lateral meniscus, and all those other accessory way stations in the auditory system. And we have ways into the supracollicus, primarily visual, but we'll forget about that for the moment, because predominant so far we've considered the other afferents of the detector. Small and medium fiber systems. On the way to the thalamus, between rights and collaterals, into stratum and meniscus. And then the output systems of the colliculus, tectospinal, which is what we started with here. And I want to make sure that it comes into the medulla in such a way that we have the MLF, tectospinal, meniscus, right next to the midline, dorsal ventral. so in half an hour. That's sort of the whole spinal cord and brain stem. Shift over into almost entirely new area, and that's the forebrain. We'll go back to our embryo. This is the forebrain, midbrain, hindbrain, and spinal cord. It's looking like this. Sulcus limitans coming up, dividing the segmental structures in the motor and association plate with the midbrain, hindbrain, terminal segments, 3 hindbrain segments, like that, with the